I'm Dr. Geneviève Bernard. I'm a pediatric neurologist and a clinician scientist at the McGill University Health Center and the Research Institute. And I'm also the chair of the Medical and Scientific Advisory Board of the United Leukodystrophy Foundation. And so I will give you a tour of the scientific symposium session. So my disclosure, I did some consultancy work for clinical trial design for Passage Bio and Ionis. I received unrestricted educational grant from Takeda. I'm a site PI on several sponsored study, including the Alexander disease study, the metachromatic. I was on the metachromatic leukodystrophy trials from Takeda. I'm on 2 studies from Passage Bio, and I'm also I was on that and on natural history studies, Kerbal GM1 and adrenoleukodystrophy. I'm also a site sub-investigator on 2 MPS2 studies. And none of these disclosures affect what I'm going to tell you today. My main disclosure is actually that I'm not an expert in all of these diseases, so I may not be able to answer your questions, and so I apologize in advance for this. So I thought that we would go over some basic genetics because a lot of these talks talk about genetic concepts. So in our cell, we have cells we have a nucleus, and in the nucleus we have the chromosomes. We receive 1 chromosome from our father, 1 from our mother. And then when you read the letters of the chromosome, you can read the DNA, and that's how you can see if there is a variant that can cause disease. DNA is transcribed into RNA, which is translated into protein. There are different modes of inheritance for diseases. Autosomal dominant is one of these forms, where one parent carry a disease variant and has 50% chance of giving this disease variant to a child, which will be affected. Autosomal recessive form of inheritance require 2 disease variants in the child. So both parents carry 1 variant and have 50% chance of passing it on to their child. So every pregnancy has 25% chance of recurrence. De novo inheritance is also dominant, but in that case, the variants happen during conception. There are other modes of inheritance, such as X-linked and mitochondrial. So this is the brain. Myelin is, so you see the cortex is the gray areas, and then the white area is the white matter or myelin. Oligodendrocytes are the cells that are making myelin in the brain. And then, so what they do is that they produce the myelin sheet, which wraps around the neurons and then has to be compacted. And that allows to protect the neuron and also for fast, there's a problem? Share what? I am sharing my slides.
Yeah, she's sharing. It's not showing.
Okay. Let me just give it a second. What about now? I told you they should hire me as a tech. Good. So I apologize for those who missed the first few slides. So we were saying myelin protects the neurons, allows for rapid conduction of the electricity in the neurons so that they can talk to each other and to the muscles, for example. So you can compare a neurons and the myelin sheet to an electric wire, where the plastic around the wire is actually the myelin. And I don't know it's not going forward. Oh, okay. So 1 oligodendrocyte is responsible for myelination of several axons. Okay. So the first talk I will summarize very briefly to you is the talk from Mayo Weinderknapp. She presented on a new leukodystrophy, so leukodystrophy with calcification caused by de novo activating Notch1 variant. So this is a de novo inheritance, so the one that we saw on the far right side. So they described 7 patients with a similar disease with an unknown diagnosis. They had progressive motor, cognitive, and behavioral difficulties. They had a very variable age of onset. Both CT and MRI showed white matter abnormalities and calcification. So that's an example of the first 4 on the left are a CT scan. So you see the brain is gray. The black, oh, can I take the microphone like this? Do this. Bon. Because I can point to things. I like to point. I don't have a pointer. I'll use my fingers. Oh, it is on. Thank you, Donna. So, it's going to be much easier like this. Okay, so these are CT scans. Okay, so they give nice pictures of the brain, but never as nice as the MRIs. So you see the gray matter and the white matter are here, and then here the black areas are the ventricles. So around the brain and in the middle of the brain we have fluids. It's called cerebrospinal fluid, and it protects the brain when you shake your head so that you don't have a head trauma. So these white area here are not supposed to be there. They're calcification. And then when you look at the brain of the patient, so normal brain white matter on this specific sequence is dark. So here it's white. These are the calcification. They're black here. And the gray matter is light gray, so there it's normal, but the white matter signal is not normal. So that's what they showed. And I'm going to turn this off, just in case I have to point again. Okay, so they did whole exome sequencing on their patients, and they identified the novel variant in Notch1, which increased the function of this protein, and that led to chronic inflammation in the brain, and that could be also seen in the CSF, so the cerebrospinal fluid. They did an autopsy on patient, and they showed the calcifying microangiopathy, so the vessels had calcium in them, the small vessels of the brain, and they thought that that was probably due to chronic inflammation, and that in secondarily led to a white matter damage. And then the conclusion is also that there's the disease mechanism for this disorder is shared with some similarities to Aicardi-Gouttière syndrome. So then Roger Min from Amsterdam presented on 2 new megalencephalic leukoencephalopathy with subcortical cyst. We also call it MLC because it's shorter. So there are 2 genes that are known to cause this disease, MLC1 and glial-CAM. MLC1 is always autosomal recessive, so you need 2 disease variants to have the disease. Glial-CAM can be either recessive or dominant, so sometimes you have 1 variant versus 2 variants that cause the disease. But so most patients have an MLC1 mutations. A smaller proportion had glial-CAM mutation, but there is a very small proportion of the MLC community that did not have a diagnosis. So in those 2 genes, so they found 2 new genes. The first is called AQP4, which codes for aquaporin 4. It's important for maintaining water. It's a main water channel in the brain, and they reported 2 patients that had a remitting form of MLC. So there are 2 different forms of MLC. There's a typical form, which has a very slowly progressive course, but there's also another form that looks very similar to the first form, that initially is very similar, but then the patients improve over time. So these 2 patients had this remitting MLC form. Then they also found variants in the gene GPRC5B, which is another receptor of unknown function. They had 3 patients, and these patients had the classic MLC presentation, so the slowly progressive disease. And then we know that this gene interacts with MLC1 and glial-CAM. So we don't know, it's been many years that we know that MLC1 causes MLC, but the function of the protein is not well understood. So we are very hopeful that this GPRC5B would be targetable with drugs. And then, but MLC Family Symposium has passed, but hopefully those who were interested were able to attend. And then the next talk was on Aicardi-Gouttière syndrome. So this is a study done by Marcus Offer from Charles Perkins Center in Australia. So he was interested in looking at blood vessels, the role of blood vessels in Aicardi-Gouttière. So blood vessels of the brain form and respond differently in AGS compared to patients that don't have AGS, and that they have a microvasculopathy, so a little bit like we saw for the other disease described by Mario van der Knap. And so what they found is that these atypical blood vessels are a feature of the disease, but they also contribute to disease development. One potential impact of their work is that these blood vessels could be a good target for future treatment development. As unlike the brain tissue, they are more easily accessible by drugs. The next presentation was on Cadazil by Fanny Elani from the Icahn School of Medicine at Mount Sinai in New York. So her lab works in a very uses a very complete approach to try to understand this disease from bedside to bench and to bedside. So they look at linking blood proteins and cells to clinical abnormalities. So they look at biomarkers. They also have cellular models of the disease, and they test treatment on these disease. They have human 3D brain in a dish. So these are organoids. So you take induced pluripotent stem cells, and then you create what we call mini brain. They're not really mini brain, but there's a lump of different CNS cells, brain cells. So you can study these cells, but you can also study the interactions between these cells. And they have other models that they're studying, and the goal is to develop new clinical trials, new drugs, and then clinical trials. And so, so she wanted me to share with you what they do to discover new treatment for Cadazil. So they're analyzing blood and brain tissue to identify dysfunction of the immune system and blood vessels. They're building statistical models to test whether these abnormalities are linked with brain changes and clinical symptoms. And they use computational methods to predict which drugs could help reverse these abnormalities. They're building cellular models in a dish, so that the mini brain are the organoids, to capture disease and compare these to data obtained from blood and brain to make models useful for drug screening. And in the future, they plan to screen a library of FDA-approved drugs and novel molecules in human dish models prior to human clinical trials, and design of clinical trials for testing and of treatment in those at risk or affected with Cadazil. So the next presentation was by Dr. Sevaga Morty, and it was titled Retrospective Natural History Study in Pfizer-Merzbacher Disease Approach and Findings. So she described in details in her presentation the methodological approach to designing these clinical trials, but I thought that probably wasn't the most it was a complex. There was a very complex diagram, so I kind of left it out. And then I summarized her findings. So there's the cohort includes 150 subjects. 36% completed. They have completely extracted the data, and 56.7 partially extracted data. So this is an ongoing project. They're a cohort from all over the world. 44.6% were US patients. Yes. So it is a REDCap database where clinicians around the world that I see, like a few clinicians around the world, including our group, input data on the patients. So when the disease started, what the mutations are, how the disease evolved, like when the child had an NG tube, for example, placed in, or gastrostomy. So this is the so we import, like we put lots of data into a database, and extraction takes the data that you want to analyze from the database. You're welcome. So 55, so a little bit more than half of the patients were international patients. About a third of the patients had an early onset between birth and 1 month of age, and the remaining 2/3 had an onset between 1 month to 1 year of age. Overall, the study cohort was very affected with significant disabilities. Acquisition of developmental milestones was severely impacted. The primary findings corroborate with our current understanding of PMD. So that was expected. This is a retrospective study. Sorry, including that, sorry, I'm gonna, hopefully, I hope I didn't cough too hard for the poor people on Zoom. So including a 15-year-long prospective natural history study performed in China, in the Chinese cohort with PMD. And that suggests that the descriptive approach with retrospective natural history study has the potential to, oh, thank you so much, enhance our understanding of the impact of PMD across lifespan. Can be hopefully leveraged as a control arm in upcoming clinical trials and enable comparison across leukodystrophies. So this is a good example of how much we can do with medical records and going back in time and analyzes the data in those medical records to describe natural history studies. The next presentation was from Julia McIntosh. She's in my lab. So her title was Polar Tree-Related Leukodystrophy Disease Modeling in Vitro. Oligodendrocyte development is altered in a reduced RNA polymerase state. So what she did is that she took mice brains and she isolated the OPC, the oligodendrocyte progenitor cells. So I told you that the myelinating cells in the brain are the oligodendrocytes. So they're born as baby oligodendrocytes, so oligodendrocyte progenitor cells, and then they have to go on. They have to so they're born in places in the brain, and then they have to migrate to where they have to be. They have to proliferate. They have to differentiate into oligodendrocytes, and then they have to myelinate. So there are very important steps into myelination. So what she did is she took these mice OPC and she used a technique with the siRNA, and then she knocked down those genes. So what she did is that she made them less active, a little bit like we see in patients. And then she looked at at all of these steps of oligodendrocyte lives. So what she showed, and this paper has been published, is that there's a abnormal proliferation of the migration is normal. There's abnormal proliferation, but what's more striking was an abnormal maturation of the oligodendrocytes and an abnormal myelination. So there's a maturation block. The oligodendrocytes remain immature. So that's probably why they don't myelinate normally. Just making sure I said everything. Okay. And then the next talk was Dr. Jude Kopper from Rotterdam, the Netherlands. So he presented on newborn screening for CTX, considerations toward implementation. So CTX is a slowly progressive leukodystrophy with a broad clinical spectrum. Non-neurological phenotypes exist. Approximately 10% of patients, that's the estimate. It's probably an underestimation. Effect of treatment with chenodeoxycholic acid depends on the age the treatment was started. So symptoms can be partially reversible if it started at an early age, and can be irreversible and progressive if it started at a later age. Symptoms onset can be prevented if treatment is started before the onset of symptoms. So CTX is a very good study, very good disorder to include in newborn screenings, because there's something we can do we can treat the patients before they develop symptoms. So he described 3 validated methods for CTX newborn screening. And then he also talked about what needs to be done before it can be implemented. So they need a case definition. Should non-neurological CTX be included and treated? Large clinical heterogeneity amongst the cohorts. When should treatment be started after confirmation of the diagnosis? Should we watch and wait before we start treating? So these are important questions, especially considering that some patients don't have neurological manifestations, so probably would never require treatment. So you don't want to give a treatment that may potentially have side effect to a patient that will never need it. So I think these questions are things that he's going to tackle now. And then he's also working on setting up an international patient registry for CTX that will help the community further delineate CTX disease spectrum, establish possible genotype-phenotype correlation. So genotype-phenotype correlation, for those who don't know, is you look at the exact disease variant that the patient has and how they're doing clinically, and then you try to see if you have 2 or 3 or 4 or 7 or patients with the same disease variant, if they're the same clinically, if they're very similar clinically. So for example, we know that in certain diseases, there are very severe mutations leading to, unfortunately, a severe disease versus mild mutation that leads to mild diseases. So this is a genotype-phenotype correlation. And that's important because you can predict if you have very good genotype-phenotype correlation, you can predict what's going to happen to the patient over time. And that's also important. It's important for counseling the families. It's important for starting treatment when treatment is available. And it's, I think I have one more thing. Perhaps it's going to come back. It's just, and then he also wished to establish and refine criteria for the implementation of newborn screening for CTX. So then we had a talk from Dr. Allen from the University of Minnesota. And then he worked on the structural insight into variant chain fatty acid transport by human ABCD1 mutation. So this is a structural study on the adrenoleukodystrophy mutated protein. So his conclusion are that high-resolution cryogenic electron microscopy has provided detailed images of the 3-dimensional structures of the adrenoleukodystrophy protein, a fatty acid transporting biomolecule associated with X-linked adrenoleukodystrophy. And then studying these images will enhance our comprehension of the biological function of adrenoleukodystrophy and facilitate the development of improved diagnosis and clinical tools for treatments of XALD. The next presentation was by Dr. Josh Bankowski. I'm just making sure I stay on time, sorry. Healthcare utilization, burden of illness and mortality in adults with adrenomyeloneuropathy. So that's what he looked at, and the conclusion of his study was that AMN imposes a substantial and under-recognized burden in men. These affected men are at higher risk of death. There is earlier age of death compared to the healthy population. And there's also higher rates of other diseases such as liver, heart, cancers, kidneys, and lung disease. Patients with ALD has more frequent hospitalization and longer hospitalizations compared to healthy patients, healthy individuals. And the cost, their cost is 15 times higher each year. The next presentation was by Joe Hacia, and he presented the mouse peroxisome research resource. And this is done in collaboration with Dr. Zuberi and Dr. Braverman. And so what the goal of their mouse peroxisome research resource is to promote basic and translational research in peroxisome biology, specifically focusing on gene encoding for peroxisomal protein. Their specific aim 1 is to consolidate, phenotype, distribute, and cryopreserve existing mouse models for peroxisome research. They also want to develop, phenotype, distribute, and cryopreserve novel mouse models for peroxisome research. And they also aim at developing a monoclonal antibody resource to maximize the impacts of MPRR mouse model. And then the next presentation was by Orna Elwar-Stein lab. And then she presented a vanishing white matter molecular overview and updates from her lab. So this is a little small, I apologize. So giving that EIF2B is a master regulator of protein synthesis, we use our expertise in the field of global translation regulation to identify molecular networks that are mostly affected in EIF2B mutations. And for that, they use an omic approach. And then using a proteomic approach to study the process of remyelination within the brain of vanishing white matter mice, they discovered a fundamental defect in the mitochondrial molecular machinery that is responsible for energy production. And that was back in 2015. And 2017 and 2019, they found that this fundamental effect also exists in isolated fibroblasts, OPCs, and astrocytes. Again, around the same time, they identified the cellular compensatory response and discovered the ability to completely overcome the defect in fibroblasts, but not in OPC or astrocytes. Given that astrocytes are central to the disease, they focus mainly on astrocytes isolated from brains of their mice. And then by analysis of these astrocytes, they revealed important insight in their energy status, compensatory reactions, metabolic problems, and hypersensitivity to oxidative stress. And then to understand this better, the translational regulation defect in these astrocytes, they developed a unique methodology that enabled for the first time the efficient extraction of ribosome from astrocytes. So ribosomes are important to start from the messenger RNA and do the proteins. This was not possible before because there are technical barriers associated specifically with doing that in astrocyte, and that these are things that they were able to overcome. So, ribosomes are the cellular machine responsible for mRNA translation into protein, and their new methodology allowed for successful application of using omics on astrocytes to look at the expression strategy of each gene in response to cytokine-mediated activation. And then they discovered that using this omic approach, they discovered that gene groups whose activation is based on manipulation of protein synthesis efficiency, and then their data show that malfunction of mRNA translation regulation in astrocytes. And then they found that one of the groups that was affected related to energy production in the mitochondria, similar to what they had seen in 2015. So the next talk was by Amy Waldman from CHOP on Alexander disease. And so she had a very good news to say that her research program in Alexander disease has grown. So the NIH is funding a natural history study on Alexander disease through the Global Leukodystrophy Initiative. There's several sites involved in that study across the US. And then for the patient with Alexander disease, she's encouraging you to get involved. And then what they do is the evaluation of outcome metrics in Alexander disease study is ongoing at CHOP. And they perform a variety of tests to measure gross motor, such as movements and leg movements and walking, fine motors, so the hand skills, speech, communication, and swallowing skills. And then they also collect MRI data, CSF, so cerebrospinal fluid, from spinal taps and blood at CHOP, but or any other glia site. And if you're interested, then they would be very happy to include them in their study. And these are annual research visits. And then for patients that are unable to travel, they have a phone study as well. And then you can contribute to our mission of understanding Alexander disease without traveling to CHOP. And then they're very committed to clinical care. So the Leukodystrophy Center of Excellence at CHOP and the clinical partner to GLIA and around the world are committed to optimizing patients with Alexander's disease health. And then they recommend a multidisciplinary team to address the various difficulties patients with Alexander disease experience. And then she's inviting everyone to the Alexander disease trials at CHOP. And then coming soon, they're hoping to share their research results on once published in medical literature later this year. And highlights include developmental outcome, GFAP levels. So Alexander disease is caused by disease variant in GFAP. This gene codes for the protein that's called GFAP. And so they measure the GFAP level in the spinal fluid and blood, and they are using this as a biomarker. And then they also look at the swallowing function as previously mentioned. And then I think that's maybe the last talk. Antisense oligonucleotide therapy in leukodystrophies. So genetic information is kept in DNA, which is then transcribed to RNA and then translated into proteins, which are the building blocks of our cells. A possible therapy in genetic diseases is to correct the RNA. And ASOs are one type of RNA therapy, and they are able to either reduce the level of toxic protein by inhibiting the RNA, the mRNA before it's translated into protein, increase the level of a functional protein, or modify the structures of an impaired protein to improve function. And then they worked on developing ASO for LBSL, and they showed that they can restore a gene expression in LBSL and reduce the lactate level in LBSL-derived cells. They also worked on developing ASO for adrenoleukodystrophy, and they showed that their ASO are able to increase the level of ABCD2, which is another gene that's very similar to ABCD1. And then they're hoping that by increasing the production of ABCD2, they can help with the phenotypes. And that they were able to show that increasing ABCD2 reduce the level of very long chain fatty acid. And that's it. Thank you. So I welcome any questions. I'm not sure I'll be able to answer, but I'll do my best.
I don't know if it's necessarily about the scientific summaries, but I've been talking to a couple patients in the course of this weekend, and there are a couple patients who it's an autosomal recessive disease, but there's carriers that seem to have symptoms and be impacted. Is there any research being done on that particular side? Is there maybe like a in an autosomal recessive, if they get the more severe, carrier gene, does that potentially have breakthrough side effects?
So it's hard to talk about this like in general, but we know, for example, that there are diseases that are dominant. If you inherit 2 copies of the dominant gene, then you have a more severe disease. That's one option. There are dominant diseases that are incomplete penetrance. That means that, for example, the parent is completely normal and the child is affected. And then we know about X-linked disease that the boys are affected because you get one, like boys have an X and a Y and girls have two X's. So we know about X-linked recessive diseases where the mother can have some symptoms as well. In terms of recessive diseases, where when you have one variant, one variant only, I'm not sure I can think of any disease that the patients have symptoms, but it may just be me at this exact moment that I don't think of it. I can't think of anyone. But what I would recommend to do is to reach out to the expert of this specific disease to discuss this and see if there is anything that can be done in terms of research. Because I think if it is the case, it needs to be looked at clearly.
Hi. Hey, for those of us across the leukodystrophies anxious to see more treatments discovered, and any themes, sort of big picture themes that from this year's conference, like, new areas of research that appear promising, or common problems that someone has found a good way to solve, or new discoveries that have the ability to sort of help others?
Yeah, so I think overall, you can see that this conference was about bedside to bench, back to bedside. There were gene discovery, so Meyer van der Knap's presentation, for example, which is the first step into, and then also Roger Min that presented new genes. This is the first step to try to understand the disease, so that's the bench. And there were a bunch of talks on the bench part of it, try to understand what the disease happens, how the disease happened, and how to develop therapies based on different types of models. And then there were also, and then go back to bedside, and you have to to do clinical trials, and you have to be able to prepare those clinical trials. So one good example of preparing for clinical trials was the PMD study, the Pfizer-Merzbacher disease, that retrospective natural history study. And so their methodology that they develop is what we all use with some variation in all diseases right now. And it was, what I like about this study is that they compare their result with a prospective study, which is the gold standard of a natural history study, is that you look at the patient on day one, and then you follow them throughout the course of their disease, and you do specific assessment at different time points. But what's interesting about this specific disease is that they showed that looking prospectively, so starting at point 0 and then going on, shows very similar result than when you're there and you have to go back with the medical records. But does that answer your question? It looks like it's all different talks on different diseases, but we do learn from each other, and we get ideas of what we should do next. And so I think, and then we also have all of these informal discussions amongst ourselves, and establish new collaborations throughout these meetings. So these are very, very important meetings for us scientists.
This is probably a very non-scientific question. We've been hearing a lot about upcoming gene therapies, and of course we have drug trials going on with guanabens or other ones. Would you be able to, in a very layman term, maybe give some information on pros and cons of the differences between the two therapies? And is gene therapy going to be a dominant form of treating diseases in the future, especially with leukodystrophies? Any comments on those?
So there's different types of gene therapies, and it's interesting because we're just finishing our work on a brochure for families to explain in layman term what gene therapies are and other and the RNA therapies. So this is a difficult question. I think there are several clinical trials right now with gene therapy. The risks are not zero, so there are significant risks. So for very slowly progressive diseases, the risk of doing a gene therapy trial at this point is higher. Like any trial with a potential drug or therapies that may lead to side effect, you really have always to weigh the risk versus the benefit. So if you have a very rapidly progressive disease and you have a clinical trial, you have of the risks of the disease, even if they're high, they have potential benefit as well, and the disease course is rapidly progressive. So the scientific community is more, and the parents often are ready to do a study that potentially has more potential side effects. So gene therapies are not magic therapies. We don't know exactly everything about gene therapies, but there are many therapies trial currently, and there will be more. So your question about what gene therapy is, there are two main ways of doing gene therapy. One is gene addition. So this is good for diseases where the gene doesn't work enough, or gene correction. So gene correction is the CRISPR-Cas9, for example, where if you have a typo in the DNA, you correct the typo. This is not quite ready for trials, in my opinion, because when you try to correct this typo, you often cause typos or problem or elsewhere. So we call that off-target effect. So we're not there yet. Gene addition, there's several trials on this. There's two ways of doing it. We called it in vivo or ex vivo. So ex vivo is you take the bone marrow cells from the patient, you put with a virus, usually it's an antivirus, you infect the cells so that and then you put the normal copy of the gene, sometimes one time, sometimes more than one time, into the patient's cells, and then you transplant the cell back to the patient. This type of approach is in general good for disease of metabolic diseases, such as it's been done very successively with adrenoleukodystrophy, and it's been done also very nicely with metachromatic leukodystrophy. It's not good for diseases that are not metabolic diseases, because we think one of the mechanisms by which the diseases improve is by cross-correction so we think that when you do it's a bit like the same mechanism as a bone marrow transplantation, but instead of taking someone else's bone marrow with the normal gene, you take the patient bone marrow, you put the gene inside, and then you give it back to the patient. So you're not transplanting the oligodendrocytes or the astrocytes with the main cell type in the brain. You're actually putting the enzyme back into cells that probably are helping by going into the brain. Some of the white blood cell can go into the brain and then produce the enzyme and reduce the toxic products that are accumulating from a defective enzyme. The other type, the in vivo gene therapy, usually is done with an adeno-associated virus, so an AAV. It can be done, it can be given intravenously. It could be done by putting it directly into the brain. And AAV is a virus that can infect brain cells. Of course, these viruses are not viruses that give disease. We wouldn't do that to patients. So their ability to cause disease is not there. So you just put their gene, they're used as vectors. So they transport the actual gene into where you want it to be. So the AAV has an opportunity of going and add the gene into the brain cells. But you never get 100% of the cell that have the actual gene in it. So that's in vivo gene therapy. Is that a good?
There is one question on Zoom, so I will read it.
Yes, I think I can. Oh, you go ahead, read it.
Okay. Was there additional info shared about VWM, perhaps by Dr. Van der Nepp?
This year, Dr. Van der Knaap did not present on vanishing white matter, but there was a vanishing white matter session, which I believe she. I wasn't able to do it because I was doing another session at the same time, but I'm sure she updated the vanishing white matter community during that session.
Okay, thank you. There is no more questions there.
questions from the room? I like to be on time. We're on time. We have 12 minutes left. Oh, good. So I can stop sharing. And then, so thank you for those online that were there. And I hope you enjoyed the talk, for those who are here, but also those who are online. And I can leave, I guess. Take this off. Mon ordinateur va pas aimer ça. So for those who are here, does that are your expectations? Does this meeting, because it's my first time I summarize the scientific meeting, so I just wanted to know if there's things that you would want done differently. My time of posing a question, because I think I'm going to have to do this for another 2 years at least. So I want to make sure that I do as good of a job as I can. Hmm? But there was also a lot of other sessions at the same time, and you know, not most, but I think probably most of the speakers that presented on these topics are also doing the sessions for their specific diseases, so they will update the community for the. Yeah. I'm glad. I think it would be nice. I think it's nice to have an overview of what's done in the leukodystrophy community. But I think having being at your own session is also very instructive. But I'm not going to take the merit of this. Every speaker sent me one slide of their summary of what they wanted me to discuss with you. I made minor modification to their slides, so I could so I could present it my way. But I didn't do that by myself. So. Good.
I think it'd be helpful, like, what are the 3 things that very simple, like, because I think most of us don't know anything about these conditions unless they're the condition that we care about. So, like, what are the 1, 2, 3 things that are the important takeaways from this that a person who doesn't know anything about disease?
That's what we tried to do.
Yeah.
But yeah, I see what you mean. But I mean, yeah, every studies that are presented in these scientific symposium are very small. Like it's a small drop into.
Yeah, but it's hard to understand the importance without any context. So, like the PMD side, for example, I know a little about PMD, so we would like, okay, we did a retrospective natural history. Why is that important? How does that fit into the broader disease landscape? Talk a little bit about, but like, is that not been done before?
Yeah. Okay. So it's yeah. So I can try to do my best on, but it's not. I don't know. Like for example, the ABCD1 protein structure. This is outside of my area of expertise. So I don't know what was done before and what would be done tomorrow.
No, maybe that would be an ask for the person who talked, like what they, what are the.
That's what they did. That's what I asked for, what they wanted to say to the families. So, yes.
Yeah, for sure.
Varying levels from scientific, relatively illiterate, to people who are fairly competent in science, people who are
Mm-hmm.
You're very welcome. Yeah. You have a job. Perfection. So keep working.
Thank you. It was very nice of you.